All right, so this is Transcription and the Genetic Code by Daniel Voloshin.
And Reno Need. Transcription is the first step in gene expression, where genetic information stored in DNA is copied to messenger RNA, or mRNA, and this mRNA molecule serves as a template for protein synthesis. Protein synthesis uses a set of rules called the genetic code to translate nucleotide sequences into amino acids, which is later used to construct proteins. Initiation is the first step of transcription. It's where transcription factors, which are small proteins that attach to recognition sites in order to initiate a transcription process. The jobs of the transcription factors are to recruit RNA polymerase, and the job of RNA polymerase is to create a transcription bubble where the DNA is beginning to unwind, which sets it up for the next process called elongation.
All right, so elongation is the second step in transcription. It follows initiation, and in which the RNA polymerase moves along the DNA template strand in the 3 to 5 direction. And basically, the RNA polymerase adds RNA nucleotides complementary to that DNA template in the 5' to 3' direction. The exception with during elongation is that during this process, uracil is actually added in the complementary strand instead of thiamine because it's RNA, which uses that nucleotide. And basically, this process will continue until RNA polymerase reaches the termination signal. And so the final step, termination, it's actually different for prokaryotes and eukaryotes. On the prokaryote side of things, you have, first of all, row-dependent termination, in which this is just a general model, by the way, in which the row protein binds to the mRNA and moves towards RNA polymerase. Basically, when the protein catches up, it causes the polymerase and mRNA to dissociate, ending the process of transcription. On the other hand, you also have row-independent termination for prokaryotes, in which a hairpin sort of structure forms in the mRNA, and that's followed by a string of uracil nucleotides, the nucleotides exclusive to RNA. And this actually destabilizes the DNA-RNA hybrid, which causes the polymerase and mRNA to dissociate, once again ending the entire process. And then when you move into the realm of eukaryotes, we don't actually know too much, or it's much less understood, the process of termination than it is in prokaryotes. In eukaryotes, the process often involves specific sequences in the DNA or RNA that signal termination, like specific codons. And you also have something called the polyadenylation signal, and that's a sequence in the mRNA that signals for the addition of the poly A tail, which triggers the process.
All right, and transcription introduces the genetic code for the next process called translation. And basically, what a translation do is they read specific sequences on the sequence created by transcription. Each 3 nucleotides are called a codon, and each of these codons can be used to create a specific amino acid sequence. And the important thing is the genetic code is nearly universal, which means it's almost the exact same in all organisms. And as you can see, some of these codons are code for the same amino acids, which makes some of the codes redundant. And this redundancy is very important as it provides protection against mutations, or what you call silent mutations, which doesn't cause as much damage. And you can also see a start and stop codon, and those signal for the start and stop of protein synthesis in order to regulate gene expression.
And that's about it when it comes to transcription and the genetic code in the process of protein synthesis. Thanks for watching and have a great day.